All right, let's do some chapter 7. So chapter 7 is related to molecular signaling within neurons. Now you know that I think neuronal signaling is both electrical and chemical in nature, right? The type of signals that I think we've talked most about is when you've got a signal that's propagating down this axon, right? And it reaches the terminal, right? It's electrical in nature here, right? And then some neurotransmitter is released from the axon terminal and it becomes a chemical signal. And then postsynaptically, there's some maybe ion channel that's over here, right? Some sort of ionotropic receptor. And that allows ions of sodium or something like that, right, to come in here and depolarize the cell and generate another action potential, right? So we go back from an electrical signal to a chemical signal, and in the postsynaptic cell, we're back to electrical, okay? Chapter 7 is slightly different, right? It deals with more chemical signaling. And in this case, right, what we see is similar in that where we start is not that much different. We start with an electrical signal, right, where we have our same action potential that's propagating down our cell. There's a release of neurotransmitter, right? So the neurotransmitter is released. But in this case, right, instead of having an ionotropic receptor here that changes permeability of ions, like this sodium here, we have other receptors, right? We talked about G-protein coupled receptors, for example. And this chemical ligand binds, right, to this particular receptor. And we get a change that's mediated through a bunch of second messengers. So this activates, I don't know, something that's an intermediate, say target 1. And target 1 is able to then activate target 2. And target 2 activates target 3. Right? Maybe this is the final step here, okay? So for this type of signaling, right, there are really 3 main criteria that we're looking for. We're looking for a signal. So maybe we can even put that here kind of to the side. The 3 criteria of this chemical signaling that we're going to be talking about. And we'll say I see, and I want this to be intracellular, right? Because we're within neurons. Not inter, but intracellular signaling. Right? The first of those criteria is you have to have some signal. And in what we drew here, we've got that. Right? Here's our signal, the chemical ligand. We need a receptor. Well, we've got that too. And lastly, we need some sort of target. Right? And in this case, we have multiple targets, but we'll talk about target 3 because it's our final one. Okay? So this fulfills our criteria. What are these targets, right? This is still very vague. There's a lot of targets because there's a lot of different types of intracellular signaling. Sometimes these targets can be ion channels. Right? And so it's possible that we can change the behavior of an ion channel. We can make it more or less likely to open. Sometimes these targets are kinases. If you don't know what a kinase is yet, you'll learn, right? But kinases are really important signaling molecules, and they tend to activate. They tend to turn things on. Sometimes the targets can even be genes. And larger scale changes can occur when we change levels of transcription. Right? So if you've ever wondered how we make a long-term memory, right, that involves changes at the genetic level. Okay? So we'll talk about that as well. So let's deal with 3 different types of chemical signaling that we often see in neurons. Okay? The first one, and they kind of go from most common to least common, the most common is synaptic signaling. Okay? Synaptic signaling will be our focus, and an example of this are those G-protein coupled receptors that I've talked about. Right? And you can see this is sort of what I drew, that we have a signal, the neurotransmitter here. We have a release event here. We have a receptor, and we've got some target. Okay, so this fulfills our criteria. A second type of chemical signaling is paracrine signaling. Paracrine signaling is different because this is localized. Right? This signal typically only activates receptors that are nearby. Local signals. Right? That's different than in synaptic signaling, where you could have a long axon and affect cells far, far distances away. These signals are local. Okay? And so we saw something like this, right, when we talked about nitric oxide, right? Nitric oxide had the ability to signal locally. Right? Affect the cells within a range, right? But only these cells within that range. It still meets all of our criteria though, right? We have a signal, we've got receptors, and we've got target molecules, right? Signal, receptors, target molecules. Receptors on all these nearby cells. The third type of signaling is endocrine signaling. Many hormones work this way. So we won't talk about it too much. But there are times when we think of this as more neuronal, right? So when, I don't know, I guess hormones of the posterior pituitary, those are made in neurons, right? And so the way that this works is that chemical signal gets dumped into the blood. So this is going to produce long distance signaling, because the blood carries this off to any parts of the body, right? Distant cells. And as long as those cells have the receptor, right, they're able to respond to that signal. This type of signaling is typically slower. Its effects are typically longer lasting. So we're not going to spend a tremendous amount of time on this, right? This is endocrine system stuff that you might do in anatomy and physiology. We're not going to spend a ton of time on it here. Okay, so why do this? Why go through this entire process? What's the advantage? Well, the major advantage is something called amplification, right? Amplification is the idea that we start with a small signal, and the result of this can be activation in a huge way. So this small signal has a huge effect. How does this happen? Well, activation of one receptor here, right? So here's the single receptor, activates all these different G proteins, right? So if we have a G protein coupled receptor in a cell membrane, multiple G proteins can be activated by that single receptor. Okay, in each one of those G proteins, in kind of a 1-for-1 ratio, right? So no amplification, activates a molecule of adenylyl cyclase, right? So there's not any amplification, a 1-to-1 ratio here. However, this enzyme, adenylyl cyclase, can convert many, many more molecules in a step where we amplify many, many more molecules of ATP, right? It can convert ATP to this substrate, cyclic AMP. I shouldn't say substrate, I should say product. ATP is the substrate. And so one molecule might activate this one, and it might activate that one, and it might activate this one, and so on. Okay? Now, in the next step, we have no amplification, where again, we've got a 1-for-1 ratio, as each of those molecules of cyclic AMP activates a kinase. Okay? But in the very last step, again, we amplify, where one of these kinases can transfer many phosphates to target proteins, right? So this might activate this one and phosphorylate it, and this one, right? So a lot of phosphates being transferred. So we've amplified our signal. For example, right, norepinephrine, right, generates thousands of second messengers, right? So we get thousands of G proteins that might get activated here, right? If this is our norepi receptor. So thousands of second messengers, right? And these second messengers go on to activate a total of, like we're talking tens of thousands of kinases. Okay, so there's an amplification step here. By the way, you know what this looks like to me? When we talk about for the times, this looks like that the receptor hasn't very well socially distanced, right? This is kind of what we think about with infection, right? That if this person can infect all these people, right, now all these people can go on to infect more people. And so this is what we get, that all these people become infected. It just seems appropriate for the time. Now let's say a word about the types of ligand or signaling molecule itself. The book likes the word signaling molecule. I often say ligand, so you'll probably hear both, right? The first sort of class of molecules are those that are cell impermeant. Right? These molecules cannot get through cell membranes, so they must activate receptors on the cell's surface. Most signaling molecules are cell impermeant. Acetylcholine was cell impermeant. Any of those amino acid based biological or biogenic amines, those are all cell impermeant. Gases are cell permeable. Some lipid based molecules are cell permeable, and that's the next category. So these things involve, like I mentioned before, gases. Nitric oxide is an example. Lipid-based molecules. Endocannabinoids, right, are these types of molecules. The way that they work is they can easily diffuse just right through the cell membrane. Right? They don't have to have any receptor on the cell surface, and they don't even necessarily need to go through any release event. Okay? And so these molecules typically signal through intracellular receptors. And they often mediate their effects, when this complex form, they often mediate their effects by translocating into the nucleus and affecting gene transcription. And we'll say more about that when we talk about the specific types of receptors. That'll be the next thing that we mention. The third type, probably the rarest type, right? We're going to spend a lot of time on cell impermeant molecules. That's so many of them. We'll spend some time on cell permeant molecules, and then I'll just mention, because it is part of the category, what are called cell-associated molecules. Cell-associated molecules display their signal on the surface. Right, on the surface of their cell membrane. So in this case, the ligand is released across the synapse. In this case, the ligand is released and crosses membranes. In this case, the ligand is never released, right? The ligand is somehow bound to the membrane. And this is where we have to stop.